Logo: National Museum of Natural History. Smithsonian. Text: Hot topic: Environments that shape us, using chemical indicators to understand the diet and climate surrounding human evolution, featuring Christine France, Manager, Smithsonian Museum Conservation Institute Stable Isotope Mass Spectrometry Laboratory, February 20th, 11:30 a.m. to 12:30 p.m. ET. On the left is a photo of a woman using a handheld tool on a fossil. In the upper right-hand corner of the screen is the speaker's video.
Hi, everyone. Welcome to today's Hot Topic program, Environments That Shape Us, using chemical indicators to understand the diet and climate surrounding human evolution with Dr. Christine France. My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray-haired woman in a green turtleneck sweater, wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us for Hot Topics or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of your Zoom interface. We're in a webinar format, so we can't see or hear you. As you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so we can sort through as many as possible. The Q&A time really flies by. The Q&A box is also where we will share any relevant links during our program, so please keep an eye out there. We'll start with an opening presentation by our speaker, Dr. Christine France, and then I'll join her here to take your questions. During the presentation, I will also write answers to some of your questions, at least any that I can answer, as will another member of our behind-the-scenes team, Dr. Ryan McCrae. So now I'd like to go ahead and introduce our speaker for today. Dr. Christine France is a Smithsonian research scientist and the manager of the Smithsonian MCI Stable Isotope Mass Spectrometry Laboratory, which performs stable isotope analyses for all Smithsonian units. Her primary field of research is stable isotope applications to vertebrate paleontology and archaeology. She uses stable isotopes in well-preserved bones and teeth as proxies for ecological influences, diet, climate, demographics, and physiological mechanisms. Her research includes reconstructing diet, mobility, and demographics in archaeological populations through time, through stable isotope data, analyzing the preservation of isotope signals in museum collections, and examining the effect of different chemical preservation treatments on isotope values in bones and teeth. And now I'd like to welcome Christine to share her presentation.
All right, thank you very much. Let me get these slides going here.
She opens a slide presentation.
All right. Are we all set?
Text: Environments that shape us, using chemical indicators to understand the diet and climate surrounding human evolution. Christine A.M. France, Ph.D. Smithsonian National Museum of Natural History. Hot Topics Lecture Series, February 20th, 2025. Logo: Smithsonian Museum Conservation Institute.
All good, perfect.
All right, thank you. Well, thank you for having me here today. I'm very excited to be here speaking to you at this Hot Topics lecture series. Today, I'm going to talk a little bit about environments that drive early hominin evolution, and specifically how we use chemical indicators to reconstruct diets and climates to get at some of those environmental factors. I'm joining you today from the Smithsonian Museum Conservation Institute down in Suitland, Maryland.
Slide:
Okay.
Human evolution. On the left is a graphic of an ape slowly turning into a human as it walks to the right, with the stages from short, hairy, hunched ape to upright, less hairy, walking man holding a spear labeled Australopithecus afarensis, Homo habilis, Homo erectus, Homo neanderthalensis, and Homo sapiens. On the right is a map of Europe, Asia, Africa, and Australia, with blue, red, and green arrows showing migration patterns. The map text is too small to read. The red arrows go from Africa to Europe, Siberia, and Australia. The blue arrows do the same but go through Central Asia and along the Indian coastline. The green arrows go from Central Asia through the Middle East and from Spain to Africa. Text: How did we become what we are? How did we get here?
So if you're with us here today, you probably know a little bit about human evolution because you're interested in it. So I won't really belabor some of these basic ideas. Just a quick review that we know humans evolved from some sort of early primate-like ancestor, and then they moved across the globe. Most theories stem some sort of out of Africa hypothesis, moving into different regions of the world, sort of repopulating, going back and forth to different areas. There's so many questions we can ask about human evolution, so many things that we want to know, but it tends to sort of boil down to two really fundamental questions, which is how did we become what we are and how did we get here? And those are really, really broad questions.
Slide. What drives evolution? On the right is a diagram of a branching tree with slightly different birds on each branch. The birds on the bottom right branches are mostly dark gray and have large beaks. The bottom left bird is small, light brown, and has a small beak. The birds on the top right branches are medium brown with small beaks, and the birds on the top left branches have dark gray heads, light gray bodies, and small beaks. Text. Darwin's Galapagos finches. Copyright National Geographic. The Galapagos Islands appear in the bottom left, and a map of Ecuador's coastline appears in the top left with an area shaded pink. In the top right is a color portrait of Darwin, a balding man in a brown vest and jacket.
So, we're really focused on this idea of what drove evolution. So the spoiler alert answer to that is environmental change. That's a big, broad answer, but that's what it really boils down to. The driving force behind evolution is environmental change. I like to show this picture here of Charles Darwin with his finches. As you probably know, Darwin was one of the first to really articulate this idea of natural selection, whereby the environment and environmental factors really drive evolution. And of course, he first observed this in these finches with their different beak sizes and body forms and how the environment really shaped their evolution.
Slide. Hominin Weather Report. Text. It would be great if we had written records of climate and environmental change during the time of human evolution. On the right is a browned and frayed copy of the Farmer's Almanac from 1818.
Now, it would be great if we could go back to the time of early hominins and have some sort of written record of what the climate and the environment was like during this time period.
The date on the almanac changes to 1 million BCE.
But of course, we don't have a written record.
Slide. Reality. Text. Usually we do not see outward signs of ancient human habitation. On the right is a photo of an arid landscape with scraggly brush. Low mountains rise in the distance. There are red-striped rocky cliffs in the bottom right.
The reality is we get something that looks kind of like this. This is just a generic landscape in Africa, and oftentimes we really don't have any outward signs of human habitation. But if we're really lucky and we dig down in the right place, we might get something like this.
3 photos appear below the landscape. On the top left is a fossil with many indentations on an oval shape. On the top right is a partial jawbone with teeth. On the bottom is a cylindrical soil sample.
We might get some microfossils, we might get some bones or teeth, we might get ancient soils and sediments that actually existed during the time of hominin evolution.
Slide. How do we dig deeper? The photos of the jawbone and soil sample enlarge. Text. Can we go from bones, teeth, or soils to environmental information like climate and diet?
So the question then becomes, can we dig deeper? Can we take some of these remains that were actually forming in the environments that hominins lived in, and can we reconstruct information about the climate or the diet?
Arrows point down from the jawbone to a photo of a desert and from the soil sample to a photo of a strawberry plant.
The answer, of course, is yes. If the answer were no, the talk would be over and I wouldn't be here speaking to you anymore. But that's what we're going to talk about for the remainder of this conversation.
Slide. Environmental preservation. Text. Burial and fossilization preserves information. A diagram of soil layers. On the bottom left in the lowest level, 2 dinosaurs lay dead on the ground. Moving up to the right, water covers that area. Further up to the right, the dinosaur bones are now in a gray lower layer while levels of brown dirt pile above them, topped with grass and trees. Finally, at the top right, a desert tops the layers. Archaeologists dig down into the gray area and retrieve the bones. Text appears next to each layer from bottom up. Environmental information is deposited. Burial, lack of oxygen preserves materials. More burial, possible rock formation, ancient soils and bones/teeth preserved. Digging or coring to retrieve fossils and ancient soils. What survives this process, and what can we learn from whatever is left?
So first, a quick review of how things get preserved in the environment. I'm showing here this sort of cartoon image that does use dinosaurs because I didn't necessarily feel the need to show human remains. Dinosaurs did not exist during the times that hominins were evolving, but this sort of cartoon process that I'm about to illustrate does apply to any sort of animal remains. And of course, who doesn't love a good dinosaur? So you have some sort of organism or animal that has died, and it is sort of sitting there on the surface of the earth. There is other environmental deposition happening at the same time. You see some plants there, you see some sediments and soils. We get burial. Hopefully we don't have too much oxygen because that helps us preserve some of this material. We get more burial, lots of sediments piling up on top, possibly some all-out formation into rocks, and hopefully some preservation of ancient soils and bones and teeth. Then we have to find it, some sort of digging or coring to actually retrieve these fossils or ancient soils. So after all of this, the question becomes, what survives? Is there anything left that we can actually extract information and learn about these environments?
Slide. What's in a bone or a tooth? A diagram shows a whole bone on the left. A portion of it is enlarged on the right, labeled tissue structure. Osteon, Haversian system, roughly 200 micrometers. It has spongy bone on the inside, compact bone on the outside, and blood vessels. A microscopic view shows a grid of circles labeled osteonic canal. The outer layer of the bone is labeled periosteum. A portion of that section is enlarged further to the right, labeled microstructure, and shows a grid of rods at right angles to each other, labeled lamellae, roughly 7 micrometers. A portion is enlarged to show a rod made of many tubes close together, labeled collagen fiber, roughly 5 micrometers, and the many tubes are labeled collagen fibrils, roughly 50 nanometers. A portion of that is enlarged to the right, labeled nanostructure. Gray sheets with purple horizontal stripes are labeled mineralized fibrils, with collagen molecules in gray and hydroxyapatite crystals in purple. The crystals are enlarged and are rectangular, with measurements roughly 50 by 25 by 2 nanometers, and the molecules are enlarged to show a collagen triple helix, roughly 300 by 1.5 nanometers. Text: hydroxyapatite, bioapatite. Primary mineral component in bones and teeth.
Okay, so let's talk about what's left. What's in a bone or a tooth? And then we're also going to talk about soils in the next couple slides here. So this is just sort of an image demonstrating the structure of bones in particular. It also applies to teeth. If you look all the way over on the left, you have the macrostructure where you have lots of pore spaces, lots of blood vessels. If you skim over to the right of this diagram, you look at the nano and the microstructure. Bones fundamentally boil down to two primary components, plus a couple of other minor components, but mostly the two big ones are the collagen protein. This is a strong triple helix protein, and then the hydroxyapatite mineral or the bioapatite mineral. And this is what we're going to focus on for the remainder of this talk. This is the primary mineral component in bones and teeth.
Text: Ca5, open parentheses PO4, comma, CO3, close parentheses, 3, open parentheses, OH, comma, CO3, close parentheses. Ca, calcium, P, phosphorus, C, carbon, H, hydrogen, O, oxygen. Carbon and oxygen are in bold.
Here's the chemical formula. It has a lot of calcium, it has some phosphorus, it has carbon in a couple different locations in that molecular structure, it has some hydrogen, and then it has oxygen in 4 different places in that chemical structure. We're going to be focusing on the carbon and the oxygen. For the sake of time, I don't have time to talk about everything, so we're just going to focus on those 2 for now.
Text: environmental water becomes drinking water. A diagram on the right begins with environmental H2O and a rainy cloud icon. A left arrow points from that to the word oxygen, and another left arrow points from that to the crystals in the bone diagram.
All right, so where do they come from? Well, the oxygen in your bones and teeth comes from the environmental drinking water. Rains down on Earth, it becomes water that you can drink, and that is where the oxygen in your bioappetite is routed from. Sometimes a little bit the water in your food, but mostly it's the water that you drink.
A diagram appears on the lower right with icons of a plant, an acorn, and text, plants that animals eat. A left arrow points from that to the word carbon, and a left arrow points from that up to the crystal in the bone diagram.
The carbon is going to come directly from the plants that you're consuming, or if you're a strict carnivore, it's going to come from the plants that your prey items are consuming. If you're an omnivore, it's going to be a little bit of a mix. And that's how the natural environment is actually going to make it into the animal bones and teeth.
Slide. What's in a soil core? On the left is a photo of a cross-section of the ground, with grass on top, then a dark brown layer with white rocks, a medium brownish-orange layer in the middle, and a gray rocky area on the bottom.
Okay, now I mentioned soils, so let's talk about those. There are lots of different kinds of soils. There are lots of different soil profiles, but they all boil down to this kind of basic fundamental structure. And that is some sort of inorganic layer at the base of the soil. It's mostly broken up bits of rock and mineral. And then at the top, you're going to have some sort of mix of organic and inorganic layer. There will be plant detritus, probably some microbes, maybe some fungal activity, maybe some animal remains. All of this is going to be breaking down, so you're going to have biodegradation products, and this is all going to be mixed up with some of the rocks and the minerals.
A diagram appears on the right with a plant and tree icon. A left arrow points from that to the word carbon, and a left arrow points from that to the top section of the soil, which includes the dark and medium brown layers.
So the carbon in the soils is going to come directly from the plants. It's kind of a really basic relationship. The plants die, they degrade, they become incorporated into this organic layer. And that's how the natural environment is going to make it into these ancient soils.
Slide. Geochemical tracers. On the left is a diagram of a cloud icon raining onto the earth. Text. How do we know if an oxygen atom in an animal bone came from a hot arid or cooler wetter period? How do we know that a carbon atom in an ancient soil came from open grassland or closed forests?
Okay, well, that's great. So it's great that we know where the oxygen comes from. It's great that we know where the carbon comes from, but that's not enough. If we really want to understand these environments and be able to reconstruct them and sort of apply them to these ideas of evolution, we need more. We need to know if that oxygen atom came from a hot, arid, or a cool, wet period. We need to know if that carbon atom came from a particular kind of plant, maybe an open grassland or a closed forest. And we're going to do that using geochemical tracers. A tracer is just a chemical signature in some sort of natural sample that you can use to backtrack where that component atom came from.
Text. This is where stable isotopes come in.
There are many different kinds of geochemical tracers. We don't have time to talk about all of them today, so we're going to focus on stable isotopes. We have a stable isotope laboratory here at the Smithsonian. It's the laboratory that I manage, and this is a technique that we use very frequently at the Smithsonian to reconstruct environments.
Slide. What's a stable isotope? Text. Same protons, same electrons. Different neutrons, different atomic weight. Below are two diagrams of an oxygen atom. On the left is oxygen 16, denoted superscript 16O. It has 8 green and blue protons and neutrons in the middle and 8 red electrons on 2 orbits. A right arrow labeled +2 neutrons points to the second diagram, oxygen 18, denoted superscript 18O. It has 8 protons and 10 neutrons in blue and green in the middle, with 8 red electrons on 2 orbits.
All right. So some of you here might be chemists. You might have had chemistry very recently. You know a lot about it. You might already know what a stable isotope is. So just bear with me for a moment. If you haven't done chemistry in a while, let's just review. Harken back to your last chemistry class and try to remember that atoms are composed of protons, neutrons, and electrons. An isotope is just an atom that has one or more extra neutrons. I'm showing the example here of oxygen, which typically has 8 protons and 8 neutrons in the nucleus. It can handle a couple of extra neutrons. So if it's in its normal state, we have 8 plus 8, we call that an oxygen 16. If it's got those extra couple of neutrons, now we have 8 plus 10, and we call that an oxygen 18. We're also going to be talking about carbon today. That has 2 isotopes, 12 and 13. Carbon typically has 6 protons and 6 neutrons. It can take 1 extra neutron to make it a carbon 13. Now, this heavier isotope is going to behave a little bit differently in chemical reactions. It's a little bit heavier and just a little bit bigger, so it's going to behave just a little differently as that atom attempts to become incorporated into chemical bonds. And this is what we call stable isotope fractionation.
Slide, stable isotope fractionation. A diagram of a chemical reaction shows a cluster of yellow oxygen 16 and purple oxygen 18 atoms on the left, with a green arrow pointing right. Below the green arrow is an icon of a human jumping over a hurdle and the text, energetic or size hurdle, with the word hurdle in quotes.
How do isotopes behave when chemical bonds are formed and broken? And the difference in behavior between those light and heavy isotopes is where we get fractionation. So anytime you have a chemical reaction, you're going to have some kind of energetic or size hurdle that you have to get over. It takes energy to make bonds. It takes energy to break bonds. So if you have some sort of pool of starting atoms, here I'm showing oxygen.
3 oxygen 16 and 1 oxygen 18 atoms move along the green arrow. Text. End product favors one isotope over another.
As we go through this chemical reaction, generally one isotope is going to be favored over the other. Oftentimes it's the light isotope because it's a little easier to break and make bonds when you have a smaller, lighter molecule, or atom rather. But not always. Some reactions, as you will see, do favor the heavier isotope.
From a cluster of 7 oxygen 16 and 4 oxygen 18 atoms on the left, the green arrow points to 6 oxygen 16 and 1 oxygen 18 on the right.
So in the end, you're going to end up with a different pool, a different ratio of the heavy to the light isotope in your end product. And that is the process of fractionation.
Slide. Effects of aridity on water isotope fractionation. On the bottom left is a photo of water and a sandy shoreline. Yellow and purple water molecules with oxygen 16 and oxygen 18, respectively, are in the water.
Okay, why do we care about this? Because fractionation happens all the time in the natural environment. And this is how we're going to trace isotopes. Let's talk about aridity and its effect on water isotope fractionation. So you have some sort of pool of water, a drinking pool where animals can come and get some water. You're going to have H2O molecules that have the light oxygen 16, and then you're going to have some heavy molecules that have the oxygen 18.
A diagram shows more yellow than purple molecules rising from the water into a cloud icon, labeled evaporation.
Evaporation really favors the lighter isotope, the lighter water. It is a lot easier to pull a molecule out of liquid form and put it into vapor form when it's just a little bit lighter and just a little bit smaller.
Another diagram shows more purple than yellow molecules falling from a raining cloud icon, labeled precipitation.
Precipitation, on the other hand, tends to favor the heavier isotope, although both isotopes will be raining down. When you condense that water back into liquid, more of that heavy isotope is going to rain back to Earth. So in really hot, arid climates, evaporation dominates. A lot of that light water is going to be removed, and you're going to be left with more of the heavy water available for drinking.
The water and sandy shore picture reappears on the right with 4 purple and 1 yellow molecules in the water.
In cooler, wetter climates, you have more of a balance, not as much evaporation. So not as much of that light water gets removed, and a lot of it rains back to Earth anyways. So you end up with more light water available for drinking.
The water and sandy shore picture reappears on the bottom right with 2 purple and 3 yellow molecules in the water.
So now we have a separation of these isotopes based on that aridity.
Slide. Effects of plant type on carbon isotope fractionization. On the left are an orange and a blue molecule, each with a carbon and O2 inside. The orange carbon is carbon-12, and the blue carbon is carbon-13.
Right, now let's talk about carbon isotope fractionation and how this relates to plant type. So now we're going to switch gears for a second and talk about carbon dioxide, CO2. And that can have the light carbon-12 or the heavy carbon-13. And this all comes into play when we talk about photosynthesis.
A picture of a tree appears in the top middle with the text, C3 photosynthesis, trees and shrubs. A photo of grass appears in the bottom middle with the text, C4 photosynthesis, grasses. On the right, a diagram of a tree, labeled photosynthesis, shows CO2 plus water going into the leaves, the text, carbohydrate, in the leaves, and O2 coming out of the leaves.
If you're a biologist, you probably already know this. If you haven't had biology in a while, harken back to your last biology class and try to remember that photosynthesis is the process where a plant is going to take carbon dioxide and water. It's going to convert it into a carbohydrate. That's just a long chain of carbon atoms with a bunch of other stuff attached to it. And it's going to emit a little bit of oxygen. Now, there's a couple different kinds of photosynthesis. They use different pathways to get to their end carbohydrates. We call those pathways C3 and C4. There's a third one that I'm not going to talk about today. The C3 and the C4 refer to a particular length of carbon chain, a 3-carbon or a 4-carbon chain in that process. I don't have time to show the really extensive chemistry of photosynthesis, so you're just going to have to trust me on that. Just to say that the C3 and C4 do not refer to isotopes. Those are about the carbon chains in the photosynthetic process. Now, C3 plants, all trees and shrubs are C3 plants. C4 plants, most grasses are C4 plants. They use that pathway. There's a few grasses that use C3, but a lot of them are C4.
An arrow points from the orange and blue molecules up to the tree photo, and 6 orange and 2 blue molecules travel along it.
Okay, now, again, I don't have time to go through the really extensive chemistry of photosynthesis, so you're going to have to trust me on this. The C3 photosynthesis really favors the light carbon isotope. So you're going to get a lot more of the lighter CO2 molecules.
An arrow points from the orange and blue molecules down to the grass photo, and 2 orange and 6 blue molecules travel along it.
The C4 photosynthetic pathway doesn't have the ability to discriminate. It would prefer the light, but it really can't. So it ends up taking a lot more of those carbon-13 atoms.
An arrow from the tree photo points right to falling leaves, labeled organic decay. An arrow from the grass photo points right to a field of brown grass, also labeled organic decay.
Now everything dies, okay? Leaves fall, grass dies, we get organic decay. And as we talked about earlier, that then gets incorporated into the soils.
An arrow points right from the leaves to a photo of soil with 5 orange and 2 blue molecules and the text, "Soil organic matter has more light carbon."
So soils that have a lot of C3 plants over top of them are going to end up with quite a bit of that light carbon in the soil organic matter.
An arrow points right from the grass to a photo of soil with 2 orange and 5 blue molecules and the text, "Soil organic matter has more heavy carbon."
And soils that have a lot of grasses growing over top of them are going to end up with a lot of that heavier carbon in the soil profile. All right, so now we see how we're fractionating these isotopes and how we can use it a little bit to reconstruct environments.
Slide. Reporting stable isotopes. Text. Data reported as delta notation. Delta oxygen 18 equals open bracket, open parentheses, oxygen 18 divided by oxygen 16 subsample minus oxygen 18 divided by oxygen 16 substandard, close parentheses, divided by oxygen 18 divided by oxygen 16 substandard, close brackets, times 1000. Delta carbon 13 equals open bracket, open parentheses, carbon 13 divided by oxygen 12 subsample minus carbon 13 divided by carbon 12 substandard, close parentheses, divided by carbon 13 divided by carbon 12 substandard, close brackets, times 1000. Units in parts per thousand. Per mil, a percent symbol with 2 circles on the bottom. Text. Standards are internationally accepted values that all labs use in their calculations.
We're going to start talking about data in just a second, so I need to show you how we actually report these isotope values. We use something called delta notation. This is a way to express the ratio of the heavy to the light isotope. We always talk in terms of ratios, as I have been talking throughout this conversation this morning. We don't really worry too much about absolute counts. We care about that relationship between the heavy and the light. So we're going to take that ratio in our sample. We're going to subtract that ratio from some sort of internationally accepted standard. All the laboratories use the same standard. It's a set value. So this allows us to normalize all of our data so that we can cross-compare one laboratory's data to another. We multiply by 1,000 just to make that number a little bit easier to use. So that gives us a unit of parts per thousand, which is per mil. It's just like a percent, which is parts per 100, but instead we're per thousand, so we go by per mil. All right, so you're going to see this notation as we go through the rest of this talk.
Slide. Direct relationships. Two graphs appear. The left graph has bone/tooth mineral oxygen isotope values on the x-axis and meteoroic water oxygen isotope values on the y-axis. It has a line that increases steadily up to the right. An arrow points down from the graph to the photo of the jawbone and teeth. The right graph has soil carbon isotope values on the x-axis and plant carbon isotope values on the y-axis. It has a line that increases steadily up to the right. An arrow points down from the graph to the photo of the soil sample. Text. We can measure stable isotope values in bones, teeth, and soils and calculate the stable isotope values of the drinking water and plant cover.
First, though, this only works if we have direct relationships. These are cartoon graphs, okay? They're not based on real data. And this is just to demonstrate how we can go back and forth between our environmental and then our artifact, like our bone, our tooth, or our soil. So this text might be a bit hard if you're on a small screen. Over on the left panel here, I've got on the y-axis the meteoric water oxygen isotope value. So that's your drinking water. It's your drinking water isotopes. Down on the x-axis, you're looking at the bone or tooth mineral oxygen isotope value. So that's your bone or tooth bioappetite oxygen. And there is a direct relationship between those two. It's offset a little, but it's a direct relationship. So we can, in fact, measure the isotope value in a bone or a tooth, and then we can back calculate what that isotope value would have been in the drinking water, which gives us some information about the environment. Likewise, over on the right panel, here I've got on the y-axis the plant carbon isotope value, and then on the x-axis is your soil or your sediment carbon isotope value. And that is also a direct relationship. So you can, in fact, measure the carbon isotope value in the organic material of your soil, and then you can back calculate what the plant carbon isotope value was and figure out if you're dealing with C3 or C4 plants.
Slide. Isotope space. A graph has delta carbon 13 per mil, VPDB, on the x-axis from more orange carbon 12, at value -1626, on the left, to more blue carbon 13, at value -8, on the right. It has delta oxygen 18 per mil, VSMOW, on the y-axis with more yellow oxygen 16, at value -12, on the bottom, to more purple oxygen 18, at value 9, on the top.
Okay, so it helps to think about this in kind of a theoretical isotope space. So this is sort of another cartoon graph. This time I'm showing you the delta 18O value on the y-axis, that's your oxygen. And then we've got our carbon values on the x. And although this is a cartoon graph, those values are actually true to what you would find in nature. So if you're at the top of this graph, you're looking at a lot more heavy oxygen atoms. If you're somewhere in the bottom of this graph, you've got more of the light oxygen. If you're on the left of the graph, you're going to have more of the light carbon 12. If you're towards the right of the graph, you're going to have more of that heavy carbon 13.
The graph breaks into 4 quadrants. The top left is orange and purple, the top right is blue and purple, the bottom right is blue and yellow, and the bottom left is orange and yellow.
So you're going to end up in one of these kind of broad 4 regions.
She indicates the lower left.
If you're down here, if your data lands down here in the left, you're looking at very light oxygen, very light carbon. That means we've got a cool, wet environment, and we're dealing with a lot of C3 plants, so we're looking at closed forest, shrubbed environments.
She indicates the upper left.
If you're up here, okay, now we're getting a little more heavy in our oxygen, so now we're trending towards sort of a more hot, arid climate, but we're still more closed forested or shrubbed vegetation.
She indicates the lower right.
If you're down here in the bottom right, now you're getting into the heavier carbon, so that's more C4 plants. We're getting into open grasslands, but it's still pretty cool and wet.
She indicates the top right.
And if you're all the way up here in the top right corner, you've got heavy oxygen, heavy carbon, you're in open grasslands, and you're dealing with a hot, arid climate. So that's how we use these isotopes to sort of place ourselves in environmental space.
Slide. Case study. Aridity in hominin environments. The beginning of a journal article title page is shown. Journal, PNAS, article title, Aridity and Hominin Environments. Authors, Scott A. Blumenthal, et al.
Now, let's talk about some case studies. Let's talk about some real data. All right, the first case study I'm going to highlight is by Scott Blumenthal and his group. This came out in 2017, and they were looking at aridity in early hominin environments.
A map of Africa with climate displayed in brown and green appears on the right. The northern section is light brown, the central section is green, darkest in the middle, and the bottom section in Madagascar are a mixture of green and darker brown. Text. Goal. Investigate the role of climate, specifically aridity, in shaping hominin environments.
So they were really interested in investigating the role of climate, specifically aridity. In Africa, they were working in the Omo-Turkana Basin, which you can see highlighted in that little red square.
A red square appears in the central eastern portion of Africa, indicating the location of the Turkana Basin.
This is an area where we know we have really abundant preservation and evidence of early hominins, so it made sense to look at the environment in this particular area.
Text. Used modern mammal teeth from known locations and known climates as a reference dataset.
The first thing they did was they used modern mammal teeth to create a reference data set. They took modern mammals from locations where they knew exactly what the climate parameters were.
Text. Used fossils mammal teeth from several sites spanning approximately 4.4 ma to 10 ka.
And they used it to create a reference data set, which they then applied to fossil mammal teeth from several sites going all the way back to a little over 4 million years.
The graph with bone and meteoric oxygen isotope axes returns, with two dotted red lines parallel to and below the original line. Text. Evaporation sensitive, ES, versus evaporation insensitive, EI, mammals.
All right, and this all hinges on this idea of evaporation sensitive versus evaporation insensitive mammals. Here's that cartoon graph that I showed you before. So your drinking water oxygen isotopes are on that y-axis. Your bone bioappetite mineral oxygen isotope value is on the x-axis. Most animals are going to fall on that gray line.
Above the original solid gray line, pictures of a hippo, rhino, and elephant appear with the text, evaporation insensitive.
Evaporation insensitive mammals, in particular, tend to fall on that gray line. What does that mean? It means that their water isotope values in their bones that get recorded in their bones don't really change depending on what the aridity is. Why? We're not 100% sure on why that is, but it definitely has something to do with how much they drink, how often they drink, how they're able to store water in their bodies and kind of homogenize these signals.
Below the lower dotted line, pictures of a giraffe and wildebeest appear with the text, evaporation sensitive.
Then we have evaporation sensitive mammals. These are mammals that that line shifts. So as aridity increases, their relationship between their drinking water oxygen isotope values and their bone oxygen isotope values is going to drop down towards those red dotted lines.
Text. Evaporation-sensitive mammals show a different shift between their drinking water delta oxygen 18 values and their tooth enamel. This shift is more pronounced in more arid conditions.
And that is definitely going to be more pronounced in more arid conditions. So the greater the shift, the more aridity we have.
Text. Comparing the enamel delta oxygen 18 values between ES and EI mammals gives us a measure of how arid the climate was.
So by using these modern mammals, what they did is they compared the oxygen isotope values between the sensitive and the insensitive mammals, and that gave them a direct measure of just how arid the climate really was during a specific time period.
Text. Using the modern mammals for comparison, Blumenthal et al. We're able to calculate the water deficit based on comparisons between the fossil EI and ES delta oxygen 18 bioappetite mammal data. Water deficit equals difference between water loss and gain. On the right are two charts labeled climate and hominin behavior and taxa. Under climate, there are two plots side by side with water deficit, millimeters per year, on the left x-axis and temperature, Celsius, on the right x-axis, with age, MA, on the y-axis. They have horizontal box and whisker plots that cluster to the right in the water deficit and to the left in the temperature graph. The hominin behavior and taxa chart has bars from habitual bipedality at the bottom up to the Middle Stone Age and Homo sapiens at the top. Some bars overlap.
Okay, so here's some real data. All right, so they took their modern mammals, all right, for comparison. They created what they called a water deficit measure. By looking at that delta 18O value between the insensitive and the sensitive mammals, they were able to get an idea of the kind of difference between water loss and gain in that particular environment. So if you look at these panels over here on the right, you can see that panel over on the left is the water deficit, where you have the more arid conditions on the right of that panel and the less arid conditions towards the left.
Text. Evidence is weak for long-term aridification across the entire basin. Climate and ecological factors likely vary across basins. Two distinct periods of aridification are noticeable around 4 MA and 2 MA.
And they looked at these fossil mammals going all the way back a little past 4 million years. The most recent animals are on the top of the panel. The older animals are towards the bottom. And you can see those dots kind of bounce back and forth, and the evidence is pretty weak for long-term aridification across the entire basin. It looks like rather the environment was kind of flip-flopping back and forth between different regimes of aridity. And we do see a couple distinct periods of aridity. They noted them just past 4 million years and then around 2 million years.
She circles 3 dots on the right side of the water deficit chart. Text. Overall, early hominins experienced periodic arid conditions. Supports idea that biological and behavioral changes may be related to the thermophysiological challenges associated with surviving periodically arid conditions and high heat loads.
But in general, this data is showing us that early hominins really experienced periodic arid conditions. And their paper states that it is likely that the biological changes that are observed were likely due to this need to survive these periodical arid conditions and these periodical high heat loads.
Slide. Case study 2 tree cover in hominin environments. The beginning of a journal article from the Journal of Human Evolution is shown. Article title. Modern African Ecosystems as Landscape Scale Analogues for Reconstructing Woody Cover and Early Hominin Environments. Authors, Anku W. Nagash, et al.
All right, next case study we're going to cover here deals with tree cover or woody cover. This is by Anku Nagash and her team. This came out just last year in 2024.
The map of Africa returns on the right. Text. Goal. Investigate the distribution of woody cover in early hominin environments.
All right, they were specifically looking at the amount of tree or woody cover, again, working in Africa.
Text. Used modern soil samples from known locations with known vegetation distribution as a reference dataset.
They did a similar thing where they used modern samples, in this case soil samples, to create a modern reference dataset. So they went to areas of various areas around Eastern Africa where they knew exactly what the vegetation cover was, and they were able to create this reference data set.
2 long areas on the north and southeast coast of Africa are circled in green. The northern circle applies to the area of Ethiopia, Kenya, and Tanzania. The southern circle applies to coastal Mozambique. Text. Applied the model to ancient soils in Tay the Awash and Omo Turkana basins up to 6 MA.
They then applied this to ancient soils in a couple of different basins, the Awash and the Omo Turkana basins, and they went back to about 6 million years ago.
A graph on the left is titled Modern Analogues. It has red and blue horizontal box and whisker plots for grassland, wooded grassland, bushland/shrubland, woodland, and forest down the y-axis, and delta carbon-13 SOM from -25 to -15 on the x-axis. The red are labeled African and the blue, non-African. Red begins far to the right and moves left as it gets lower, remaining roughly the same width. Blue begins in the middle and moves slightly left as it goes lower, getting narrower and narrower. Text. The delta carbon-13 values of the soil organic matter show clear distinction between different amounts of woody cover.
Okay, so here's the modern data. All right, so this graph here, you can see on the y-axis, it just sort of shows the different types of vegetation environments, from the closed forests on the bottom all the way up through the patchy shrublands to the grasslands at the top of that panel. The x-axis is the delta 13C value of the SOM, which is the soil organic matter or soil organic material. And you can see that there is a trend. Those bars shift towards the upper right corner, getting more and more positive, showing more and more C4 grasses as you get up towards the grassland.
Text. African ecosystems show unique trends compared to other regions.
They also looked at some non-African data, which you can see in the purple bars. And you can see that the trend is a little bit different there. And this just highlights the need to make sure that you're using modern data sets from the area that you actually intend to study your fossils, because the data could be a little bit different from other areas.
An arrow appears to the right of the graph pointing down, turning from gray at the top to green at the bottom. The top is labeled less than 10% woody cover, and passes through 10 to 40% woody cover, roughly 40% woody cover, down to over 40% woody cover. Text. Each vegetation type has a known fraction of woody cover. Create model to predict fraction of woody cover based on delta carbon-13 values of the soil organic matter.
So now they created this really nice model where they could relate the percent of woody cover directly to those carbon isotope values. So the forests generally have greater than 40% wood cover, and then we get less and less as we get all the way up to the grasslands, where we have less than 10% woody cover. So now we've got this nice model, and we can apply it to the carbon isotope values that we see from our ancient soils.
Two columns appear on the left, labeled Awash Basin and Omo-Chircana Basin. Each column consists of a series of bell curves graphed on a background that moves from green on the left to yellow on the right. The graph key is too small to read. The Awash Basin column begins at the top with low blue and red curves overlapping, moves to just low blue, then low blue on the left and high blue on the right, then blue for several layers, moving to the left, then low red on the right, low blue on the left, and high red in the middle. The Omo-Chircana Basin begins with pink on the right, then high pink and red in the middle for several layers, high pink and red on the left for several layers, then low pink on the left, then high pink moving right, left, and right again.
So here's the ancient data. All right, some of this text is a little hard to see, so I'll walk you through this. We have our two basins, the Awash and the Omo-Chircana. The most recent data is on the top, and then we go back in time to the bottom, all the way back to about 6 million years. The greenish side of the panel is more woody cover, more tree cover, more forests. The right side of those panels, the brown side, is the more grassy, open grassland cover. And they calculated the percent of woody cover based on the carbon isotope values. So each little hill, the crux of the hill, the apex of the hill, represents the most likely percentage of woody cover. And it tails out because there's always a little bit of error in every model, as there should be error in every good scientific model.
Text. Overall decrease in woody cover and a transition to more open grasslands in more recent time.
But you can see these hills, these little hills, kind of start migrating just a little bit towards the right, suggesting that as we come more and more recent in time, we get a decrease in woody cover. We start trending more towards those open grasslands.
Text. Appears that early hominins definitely utilized patchy woodland habitats, but existed in landscapes with a variety of different vegetation. The bottom half of each chart is outlined in red. The boxes then move to the top halves of the graph. Text. As hominin evolution proceeded, these early environments opened into more abundant grasslands. Diversity of habitats.
But in the time that early hominins were evolving, we're definitely down here in this region where we had patchy woodland habitats. So they definitely existed in a landscape where we have a variety of different vegetations. And then as we get more recent, we move into these more abundant grasslands. So yet another shift in vegetation type that hominins had to deal with and evolve into. So again, showing a real diversity of habitats here.
Slide. Conclusions. The graphic of the ape walking and turning into a Homo sapiens man with a spear returns on the left. A red arrow appears over the graphic pointing to the right. Text. Often we think about evolution as being driven by one main factor.
Okay, so just a few broad conclusions here to wrap things up. A lot of the times when we think about evolution, we really want to find the smoking gun. We want there to be that one main driving force that we can really point to and say, this is it. This is the thing that made us what we are.
The red arrow disappears and several green arrows angle into the graphic from above and below at each stage. Text. Environments apply many evolutionary pressures.
But environments don't work that way. They almost always apply many different types of evolutionary pressure. But that heterogeneity, that variety in and of itself is a really powerful driving force.
Text. The geochemical indicators have shown us that hominins were exposed to an ever-changing environment with a variety of challenges including fluctuating aridity and shifting vegetation. Hominins evolved to be adaptable.
That need to adapt to many different challenges is likely what drove hominins to be the form that we are today. Our geochemical indicators showed us definitely that these hominins were dealing with fluctuating aridity and shifting vegetation. So these geochemical indicators are really suggesting that hominins evolved to be adaptable. Okay, I'm going to stop there. I hope this gives you a little bit of an idea of how we can use chemistry and go back in time to reconstruct these environments and learn a lot more about how these hominins were living in these areas.
Slide. Acknowledgements. Text. Briana Pobiner, Ryan McCrea, Amanda Chandra, and the Hot Topics team. Smithsonian Museum Conservation Institute Stable Isotope Mass Spectrometry Laboratory. Blumenthal, S.A. et al. Aridity and Hominin Environments. Nagash, E.W. et al. Modern African Ecosystems as Landscape-Scale Analogues for Reconstructing Woody Cover and Early Hominin Environments.
A few quick acknowledgements to the Hot Topics team for their invitation. I'm so grateful to be here today. A nod to my group at the Smithsonian Museum Conservation Institute, and there's those references if anybody would like to jot them down and look at them. Okay, and I guess, Briana, we're ready for questions if you've got any for me.
Fantastic.
Shall I stop sharing?
Yes, that sounds great.
Briana's video takes over the screen.
Thank you very much. And we will post at least one of those links, the one to the open access paper, probably in the Q&A box. So I'm going to actually start with some questions that our behind the scenes team has been generating during your talk. So our first question is, why would absolute values of isotopes not be useful for your research? And would they be useful for any type of research? Why or why not?
As they speak, the video switches between Briana and Dr. France. Dr. France wears a green shirt and glasses, has long brown hair, and her background is blurred.
Okay, so absolute values with isotopes vary a lot by lab to lab. And if I take a sample from a bone that is just like 0.5 milligram more than another lab, I'm going to get a greater absolute count. But that's not really reflecting the environment. That's just reflecting how big my spatula is when I take my scoop of powder. I really care about that ratio because that ratio, it doesn't matter how much you take. The ratio is going to be the same from every single laboratory. If I take that same powder and send it to another lab and they take a slightly different amount, they should get the same ratio that I got. So it is yet another way to cross compare.
Excellent.
Absolute values. Absolute values are good for other tracers. So there are other geochemical tracers, like let's say we want to know the amount of lead in a bone. That matters for health reasons, right? Well, we do care about absolute counts in that case because there's a certain tolerance that the human body has, and if we go above it, that's bad for us. Doesn't matter what the isotope is, we just want to know the absolute amount of lead in that bone. So in that case, a pure absolute count would be what we would be after. So it is useful in other ways.
Got it. Okay, that's helpful. Thank
Yep.
you. So you showed sort of these four generalized like types of environments. Can isotopes be useful for thinking about seasonality in terms of those different environments? Or what kind of evidence might you need to tackle that question?
Yeah, absolutely. Seasonality is a question that a lot of isotope scientists do focus on. The way to do that is to take some natural tissue that grows in a serial way. And we do this a lot with teeth. When
Okay.
teeth are deposited, if you think about the enamel, that hard outer enamel on your teeth, it isn't deposited all at once. It's
Mm,
deposited in increments over the course of that tooth formation. So a lot of big mammals like elephants, cows, it takes them several years to deposit all of their enamel, and some teeth, most teeth kind of lock it in after a certain amount of time. But what you can do is take little tiny samples all along a transect of the tooth, and you
mhm,
can look at the oxygen isotopes, and what you're going to get is this really neat curve of the oxygen going up and down, and that's going to show you the seasonality of your really hot, dry summer versus your cooler, wetter winter and into your spring and fall. Some places don't have dramatic seasons, and it's kind of a flat line as you go
mm,
across that tooth. Um, some people will do this with shells. Shells also continue
huh,
to grow and grow and grow, and you can do serial sampling along a shell to look at seasonal variability.
cool.
Yep.
So here's a question that sort of builds on that, thinking about maybe other sources of variability. So for animals that migrate, so we think about the classic kind of Serengeti Masai Mara migration with zebras and wildebeest and gazelle, maybe even hominins in the past. How might that influence our environmental reconstructions using isotopes, or would it at all?
Right. That is an excellent question. And you do have to definitely consider the long-term migratory status of any animal. A lot of animals don't migrate all that much, so it's not
Mm-hmm.
that big of a deal. You're kind of getting a sort of broad general overview. Some animals do migrate. One way to get around that is, as I believe they did in those papers, in the tooth paper in particular, you don't take just one spot, you sort of homogenize across the entire tooth. So you sort of grind up a whole bunch of powder to capture all the seasons. And so what you're basically getting is a lifetime
Mm,
average value. And that's
okay.
one way to sort of combat the issues that we get with migration. But maybe we do care about migration. We in my lab, we work on a lot of modern animal studies where we do exactly that. We want to know where were they living at different times in their life, and that tells us about their migratory status and where they're moving. So isotopes can be a really great tracer for that, if that's the question you want to ask.
Excellent. So here's a question from Dudley who asks, does your data, or I guess maybe some of the data you presented, support the theory that increasing grasslands drove the evolution from our like common ancestor with humans and chimpanzees? And the data you discussed seem to post-date that early change.
So there's a lot of different theories surrounding that. I just presented the one study. What we can
Hmm.
say is yes, grasslands were opening up as humans were evolving. Was
Hmm.
one driving the other? That's a little bit harder to say, and I honestly
Hmm.
think we need a little more data from different locations, maybe some locations without open grasslands, some with open
Hmm.
grasslands, where we can really compare and see, are we seeing the same trends? Or are we seeing
Hmm.
different trends? So I think there's perhaps some preliminary support for that, but I think we need
Hmm.
more data before we can definitively answer that question.
Yeah, that makes sense. It's hard to, like you said in the beginning, it's hard to find those sort of like main drivers in some sense in the past. So here's a methods question from me. So you mentioned about taking samples from teeth, and I'm wondering, are samples that are taken for stable isotopes, are they always taken from teeth or the same parts of the skeleton? Is there any variation? If you like, can you take them from bones? And I've also heard about like samples for isotopes from things like ancient soils. Like, where does the sample, like where you get the sample, how does that play into your research questions, I guess?
Yeah. So in terms of bones and teeth, yes, you can use bones or teeth because the
Mm,
general structure of that sort of bioapatite collagen matrix is pretty much the same. The exception is
mhm,
enamel, which doesn't really have any collagen in it, but that's okay. The bioapatite is still a very similar structure. It does depend on where you take it. And here's why,
mhm,
because different parts of your body form at different times of your life. Animals tend to form their teeth, and we all know this from being children, right? We lose our baby teeth and then we get new teeth. But once those new teeth come in, they lock in and they stop growing. So your
mm,
teeth and a lot of animal teeth capture sort of the adolescent time period of life. Your
mhm,
bones constantly remodel. I won't say my age on this webinar, but I am old enough that my bones have probably completely remodeled by this point since the time I was young. It takes a good 10 to 20 years to completely remodel a bone.
huh,
So if you took my upper arm bone, my humerus, and you drilled into that and took a sample from that, it would record the most recent 10 to 15 years of my life. Some bones remodel even
huh,
faster, like ribs. Ribs only take about 3 to 5 years to remodel. So you can take a rib and you can get the really, really recent time period. You can also do this with hair, which we all know how fast our hair grows. It's only captured, my hair probably captures a couple years because it's pretty long. Your hair probably captures less because it's a little bit short. So depending on which bone or which tooth you're looking at, you can actually reconstruct diet and climate at different time periods of life. How is diet
mm,
changing from infancy to adulthood? And this is another really interesting question to ask. So, yes, the question matters, as you say. You really got to think
mhm,
about, what do I want to know? Do I want to know the lifetime average? Do I want to know just what was happening right before that animal died? And that sort of dictates where you're going to sample and how you're going to do your sampling.
cool. I did not know that different bones remodel at different rates.
Mhm.
That's very cool.
Yeah.
So here's a question that follows on really well from that, from someone in the audience. Urate asks, oxygen isotopes in hair have been used to trace woolly mammoth migrations. Has this technique been used in African animals?
In hair, it's definitely been used in bones and teeth.
Hmm.
Generally, actually, that's an interesting question. Yes, isotopes have been used in mammoth hair, as far as I remember. Oftentimes, actually, we do that more with hydrogen isotopes, which is not something I got a chance to talk about because hydrogen really changes with geographic location in a more extreme way than oxygen does. But you can do it with oxygen too. I know that hydrogen has been used in African samples, possibly a little bit of oxygen in hair too. I'd have to jump back into the literature and confirm that, but I know it's been done with hydrogen.
Cool.
Yeah.
Actually, I'm going to ask now another behind the scenes question, which follows on really nicely. So you talked a lot about oxygen and carbon. What other stable isotopes are there to look at? You just mentioned hydrogen. And what type of information do those other isotopes give you?
Yeah. Well, I didn't get to talk about nitrogen or hydrogen.
Hmm.
Nitrogen is in the collagen. I just left that out of the talk completely because I didn't have time to talk about it, but
Hmm.
protein has a ton of nitrogen in it. And nitrogen is a really wonderful way to look at trophic structure. So every time you consume something, our bodies like to excrete the lighter nitrogen isotopes in our waste products, and we hold on to the heavier isotopes. So as you go up in the food chain, you get more and more heavy nitrogen. So this has been used in African ecosystems for sure, where we will look at everything from the carnivores, the hyenas, down to the antelopes and the herbivores, and you can reconstruct who was eating whom,
Hmm.
figure out who was at the top of the food chain and who was all the way at the bottom of the food chain, and which animals were omnivorous, kind of falling somewhere in the middle. So nitrogen is a really great way to do that when you're trying to reconstruct an entire ecosystem, or how much meat, a
Hmm.
hominin was eating. Were they heavily carnivorous? Were they more omnivorous? Things like that. Hydrogen, as I just mentioned, it operates similarly to oxygen in that it's very dependent on the water, so the temperature, the evaporation. We get hydrogen through the drinking water, just like we do the oxygen, so there's a lot of similarities there. It's just that hydrogen has a much bigger range of isotope values, so sometimes you can see it more clearly when you're dealing with a shorter range migration. So you can see that in the hydrogen where you might not be able to see it in the oxygen quite as well. And we do that a lot in my lab using birds. We do a lot of bird migration studies in our
Oh, cool.
laboratory, looking at how they migrate through North America and other places.
And what about strontium? That's another isotope system I've heard about with hominins.
Yes, so strontium is another one. It functions a little bit differently. Strontium is a heavy metal. Okay, so instead of a light isotope, now we're dealing with heavy metals, which kind of move through your body in a little bit of a different way.
Okay.
But you get your strontium from the food you eat. It enters the food system through the plants and through the soils, which all come from the geology. So the underlying bedrock geology weathers away and strontium is released into the groundwater, which is then taken up by plants, which you then eat and become part of your body. And that strontium sits in the calcium site in your bioappetite. So strontium is directly related to the geology. Different types of rocks have different strontium isotope ratios. So if you know what your geologic underlying bedrock looks like, you can use the strontium in animals to kind of map out their geographic location. This works for broad geology and broad geographic location. It's not going to work, say, if I just want to know within a few miles, that's not going to work too well for me. So it has to be kind of on a broader scale. But yes, it has absolutely been done with hominins, I believe.
Okay. Here's a question from Anne from the audience who asks, can you tell what hominins were eating?
To a point. Okay, to a point. So I talked a little bit about nitrogen. So you can get a sense of how much animal protein they had in their diet, a lot or little, and it's relative, right? So you want to measure the hominin diet, and then you would want to compare it to, say, some hyenas or some real hypercarnivores in that environment and compare it to some herbivores to sort of get a sense for where they fall. You can also use the carbon to look at what kind of plants they were eating. We get into this more as humans started to cultivate their own plants and started to kind of keep their own little gardens and stuff. Certain plants use the C3 photosynthetic pathway a lot, and this is particular plants like wheat. But a lot of the grass family, as I mentioned, uses C4, and that includes things like corn and millet. So when humans really got into like cultivating their own corn and their own maize, we start to see that signal, that C4 signal showing up in their bone carbon.
Nice.
Yep.
Here's another one of the behind the scenes questions. Would herbivores and carnivores give different results if you're using isotopes to reconstruct environments? And how might you sort of standardize your measurements to account for that?
Okay. So that's a great question because it gets into the idea that carnivores kind of have this extra level of processing, right? So the herbivores directly eat the plants and drink the water, but then the carnivores eat them and they have to process like an extra level of processing there. Typically, we do sometimes see a little bit of strangeness and not as good of a direct relationship with the carnivores to the drinking water. They do get some fluids from the meat they eat. So sometimes they don't drink as much as herbivores do. Depends on the carnivore. So you have to be a little careful. You got to know your carnivore. You gotta know the physiology a little bit and understand what their relationship is to their drinking water. We tend to do it mostly with herbivores because they have nice big teeth. A lot of them have nice big bones and they're plentiful. There's a lot of them in the fossil record. Carnivores are more rare in the fossil record. And when you do this, you need more than one. You need multiples, you need replicates in order to make sure that you're really getting an accurate value. So you could use carnivores, but herbivores are better. So we generally stick to the herbivores.
That makes sense. So here's maybe this is another behind the scenes question, maybe another complication or wrench. So what about aquatic or amphibian or marine animals? Does that change how isotopes capture the environment, or doesn't it?
Yeah, it goes right out the window.
Excellent.
Yeah,
Okay.
yeah. So, when you're living in a body of water that is constantly circulating and mixing, you don't get that same oxygen seasonal variability that you do with terrestrial mammals for sure. So this would not work very well with a marine or a fish. Maybe a little. Now, I didn't get a chance to talk about how oxygen fractionates in different temperatures. So cold-blooded animals get interesting because
Huh.
cold-blooded animals do some fun things with oxygen fractionation depending on the temperature that their bones are mineralizing. That's not true with mammals because we're homeotherms and we maintain a very tight temperature range. But some fish and other, like we've done this in organisms that live in lakes, like terrestrial lakes and stuff like
Yeah.
you can actually use their bones to some extent to reconstruct the temperature variation, not so much the aridity, but the temperature variation. So there's some neat stuff in those kinds of studies to be found as well.
Oh, nice.
Yep.
So Urate has another question. Has fossil poop been suggested to the same kind of analysis that bones have?
Mm, Sure. The one challenge with fossil poop or coprolites is that they often start to mineralize. So they interact with the groundwater and they start to change in terms of their composition. That's a problem because if it's not original anymore, we're not getting an original signature. However, some coprolites have been explored that if they incorporate bits of bone chunks in them, you can extract the bone chunks, and if they're still in good shape, you can use those in a similar way. The organic material, if it's not fossilized and it's not too badly altered, sometimes you can pull organic material or like little bits of plants and stuff from it. The challenge is knowing who the coprolite came from.
Right.
So if you don't know, it kind of starts to all look the same. So you kind of have to understand a little bit about what animal deposited that coprolite in order to kind of backtrack and understand the context. Possible, but there's some additional things you need to be able to figure out.
Okay.
Mhm.
So here's kind of a broad question from Steve, who asks, what does the clustering of early human occupation sites, stable isotopic data, and plant and animal fossils show the role of ecosystem diversity and mosaic landscapes was in human evolution?
That's a loaded question that I'm not sure I'm entirely qualified to answer that because that draws in a lot of different fields.
hmm
I think I will cheat my way out of that answer and kind of reiterate that what we're seeing from the geochemical indicators, which is what I can speak to, is
hmm
that habitats likely were patchy, that we
Okay.
have this variety of different, environmental parameters that are occurring within the same basin. So there probably definitely is some patchiness. I have to think that probably played a role. I can't off the top of my head, like draw all that data together and cite a
Yeah.
specific, example of it, but it certainly doesn't look like it was a homogeneous landscape at all. So you
hmm
have to think that they had to be able to adapt to this sort of patchy mosaic type of
mm
landscape.
hmm Yeah, that makes sense.
Mhm.
Okay, so another behind the scenes sort of method-ish question. So in your research, do the isotope values always get like sort of read easily? Do they work easily? Does the research pipeline function smoothly, or are there kind of roadblocks, methodology issues along the way?
Oh my goodness. I will say this. I love saying this. Good science is never smooth. If it is smooth, you're
Mm,
probably doing something wrong.
yeah.
Because natural environments are not simple. They are
hmm
never simple. And so this data shouldn't be simple. If it is simple, then you're missing something, right?
hmm
So the data needs to be complex in order to give you the complete story. So we'll start at the beginning. Methodological roadblocks, of course. These samples
hmm
have to be excavated. There's some extensive extraction chemistry. We have to isolate the bioapatite. We have to, if we're working with collagen, we have to isolate the collagen so it goes through days, if not weeks, of chemistry before we're even ready. Then we have the mass spectrometer, which is a complex instrument that, yeah, it can be finicky. It can break down. It cannot work. We run lots of standards, lots of quality control to make sure that the data on the back end is good data. Okay. We also have to analyze our samples in advance to make sure that they're well preserved. And that is a whole nother talk about bone chemistry and how we do that. But that all has to happen. So we have to ensure that our data is good, high quality. Once we get the data, Yeah, it's never simple. I mean, you saw on some of the data I presented that there is error connected with this. There's always going to be uncertainty. So we take that into account and then we make the conclusions that we can make best, keeping that error and that uncertainty in mind. So yeah,
Mhm.
lots of things can go wrong. It is my guess that I worked a little bit with Ankunagash and I
Mhm.
run some samples with her. She spent years getting that data,
Mhm.
years for those two little images that I showed. You know, it takes a lot.
Yeah. No, it's helpful to like understand the sort of complexity and time and effort that goes into generating these data. So
Mhm.
I think I will wrap up with one final behind the scenes question, actually. Um, so, where do you see the field of stable isotope analysis progressing in the future beyond like sampling, measuring efficiency and accuracy? Like, is there a big next frontier in this field?
Sure, sure. So I see three off the top of my head. The first is just advances in the instrumentation. It's getting better and better. It's
Okay,
getting easier to use it. It's getting more user friendly. There's even some instruments that are field deployable for water isotope measurements in
cool.
particular. Can't quite do it yet with like the bones and the teeth. Um, so that could be something really cool that we might see in the future. The second is we're moving more and more towards big data. You hear this term a lot in the media, big data, right? Metadata, big data. We are now able more and more to combine different isotope systems together and create these sort of multivariate models where you can plug in multiple variables and get an even more nuanced picture of what
Hmm.
the environment would have looked like. And this is becoming a lot more user friendly in the sense that, there are statistical programs and stuff that are a little, not entirely, but a little more plug and chug than they used to be. So it's a little bit easier to kind of input that data. Um, oh, and I had a third one that just went right out of my it just went right out of my brain. We are always exploring new isotopes as well. People are working with calcium isotopes. People are working more with phosphorus and sort of combining heavy metal analysis with stable isotopes. So just sort of getting more and more involved in that. We are about to get an instrument in my lab that's going to allow us to do something called compound specific work. So now, instead of just being able to measure, let's say, the nitrogen in the entire collagen of the bone, I'm going to actually be able to break it down to the amino acids inside that protein, which give me even more information than I could get before. So the technology is getting better, it's more accessible. So we're kind of moving in this direction, I would say, of getting more nuanced.
Hmm.
Still need the bulk analyses, but we're getting more and more nuanced with it. So I think there's some really great avenues that are going to happen in the future. I'm excited for it.
Very exciting.
Yeah.
Excellent.
Yep.
So this is a great place to wrap up, and we'll conclude today's Hot Topic program. Please join me in thanking Christine for sharing her work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae, who works with us at the Human Origins Program, to our donors, volunteers, and viewers like you, and finally to all of our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. Next month, on March 20th, we're hosting Adam Van Arsdale for a hot topic program on the scales of time and space in human evolution. So don't miss that. We've put a link in the Q&A to where you can find information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's really the best way to stay informed on upcoming programs, including the hot topics, and to learn more about the museum's research and exhibitions. After this webinar ends in just a moment, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you to Dr. France, to our participants, and to you, the audience. We'll see you next month.
Logo, National Museum of Natural History. Smithsonian.